Hi, everybody. My name is Joe Luczkovich. I'm going to give you a little brief introduction to our poster. Let me make it full screen mode. This is comparing side scan imagery and single beam sonar for benthic habitat surveys in estuarine systems in North Carolina. Now, my co-author is Noah Gwynn, who will be giving a talk later in the meeting about some of this work, specifically the ground truthing and core sampling that he has done. But the purpose of the study was to see if side scan sonar can be used to survey low salinity submerged aquatic vegetation in the Neuse River estuary, Pamlico River estuary, and Albemarle Sound. We have been using a single beam sonar method since about 2010. We use two different kinds of biosonic system, but I'm going to focus on the Lowrance Elite 9 TI Echo Sounder, which is a recreational grade sonar system. Buy it at any retailer that sells fishing equipment and boating equipment. It's got a built-in side scan sonar, as well as a down viewing single beam sonar. The two types of sonar then are collected simultaneously along the same transect line. We use two different kinds of sampling methods. One where we followed the shoreline at 1 meter depth, like in Kitty Hawk Bay, shown here, and collected both the single beam sonar, and when we crossed into grass beds, it looked like this picture down here, where you see a ragged sort of canopy of grass above the bottom. Off to the side, however, the side scan sonar was also collecting out to 12 meters on either side of the boat, a picture of the bottom, an image, and you can see SAV on that as well. So the single beam's looking right below the boat, and the side scan's looking off to the side, as you see here in these two figures. The information is also recorded about your lat and long and your speed and the depth of the water. So you get accurate bathymetry from the down scan or the single beam sonar, and you get an image of what's off to the side. When you compare these two systems here on one of our along shore transects going at 1 meter water depth, we have to sometimes avoid docks as shown here and stay at 1 meter. But the company BioBase Maps will take the Lowrance single beam data and turn it into an interpolated SAV biovolume map. In the picture shown here, where it's red, there is the close to 100% biovolume of SAV. The side scan data, we use another program called ReefMaster to analyze it, and it shows also variation in the side scan data along that same exact transect. So where this is very red and indicates in the single beam sonar that there's a lot of SAV, you see a lot of SAV on the side scan as well. Then we would take the BioBase Maps output, which once you run 40 transects perpendicular to shore in an area, it gives you a nice map after you send it to the BioBase map company. They return within an hour or so a very beautiful map like that. We would take the side scan data and run it through ReefMaster and then produce a mosaic as shown here in this bottom picture, and then by hand draw polygons around areas that appeared like this that had SAV. So this is a hand-drawn method, and this is a computer algorithm that's processing the single beam data. So the side scan data takes a lot longer to analyze, but eventually you can compare the two. And what you see in these tables down here is a comparison of the number of transects out of 120 where we had SAV occurring on the single beam transect 68 of those 120 times, we also observed it on the side scan. On 6 times, it was observed on the single beam, but not the side scan. In this next line in the table 1, you see that there were places where the single beam didn't pick up any SAV. If there's no SAV, it just appears as sort of a empty bottom, a red line going across, no jagged peaks. So single beam said no, SAV was present 38 times, and side scan agreed. Our side scan interpretation, we agreed 38 times out of that. There were 8 disagreements, however, where the side scan did not detect SAV, whereas the sonar system single beam did. When you make a area estimate using the 2 different methods, using the bio-based algorithm, you get slightly higher estimates of SAV aerial coverage. The bio-based method may, with their algorithm, may actually overestimate areas of SAV because of false positives. The hand-drawn polygons, on the other hand, gets a lower estimate of SAV at each of these sentinel sites in the Neuse River, but may actually be much more conservative in terms of estimating the area, so that you can interpret those as sort of a min and a maximum of area at each site. The side scan sonar, may not really capture very small plants that the single beam sonar does. So I encourage you to read the poster more carefully and contact Noah and myself in the chat box if you have questions. There's a short video that you can watch, this video, and we'll show you some field work and sonar setup here after the end of this video. So thanks for listening, and hope to hear from you at the AERS-SEERS meeting next week. Okay, I want to show you some pictures of our sonar. This is the Lowrance and Total Scan. This is the DTX from Biosonics. We run 40 shore normal transects at a sentinel site. This is the Trent River site, and you get a map from Biobase that shows where the SAV is densest. That's the red areas on the map. And then you can do change over time. That's Edenton, North Carolina, Albemarle Sound, a 5-year period. We lost all the SAV. You get a longshore map of SAV density as well. And this is Kitty Hawk Bay. And then with Reef Master, we use the same transect to look at side scan sonar along those longshore tracks. Now some field work shots.
Where are you guys?
Albemarle Sound.
What's the name of your team?
The SAV squared.
What does that mean?
It's submerged aquatic vegetation. And then it's also, the second part's the
SONAR and video.
Team. Yeah, sonar and video team. All right. And yeah, I'm here with them. This is Dr. Joe Luczkovich, East Carolina University. Our vessel is the RV Pinfish. And we're at Albemarle Sound at Sandy Point. Say goodbye. Say, go Pirates.
Go Pirates! Arr!
That was Dan Crane and Hildy Spate, Tyler Peacock and Allie Mulligan, John Sherman, Noah Gwynn, and myself. This is the boom setup. We have a new arrangement with the metal boom, Biosonics transducer on one side and Lowrance on the other. This is a coring. After the sonar has been completed, we go and take quadrat samples. Noah will be talking about this in his oral presentation later, along transect lines that go into shallower water where the sonar cannot be used. And we get core samples with identifying plants, species composition by taking the plant samples back to the lab. This is Xanachellia being collected at the Cahuki Creek site on the tributary for the Neuse River estuary. Very low salinity. There's a lot of Rangia and other bivalve shells in the area, showing its importance as fish habitat and wildlife habitat. That we bag up the plants, then we take them back, sort them out, identify them, and dry and weigh them. We have thus species composition from every site. Here's the lab work.
Cecilia is hooking up the sonar equipment. This is a Biosonics unit, DTX echo sounder, and there are two transducers, a 400 and a 200 kilohertz transducer on that biofin. A cable gets attached to each one, and just like that, it gets attached. Okay, there's seagrass there or there's not. Actually, it all gets stored on a laptop, and you run the laptop software called SAV to do an analysis almost immediately after getting off the boat. Within 24 hours, you can have a map of the seagrasses.
So once that whole sonar rig is set up, you got the laptop running, you put the transducers on the fin in the water, and you run your transect. Last thing we do is go to 100 video verification points with a drop camera. Shown here, we visit these. These are randomly selected points in our map area, and if the bottom has no SAV, it's pretty clear. Even though the water itself is kind of turbid, so that location is sand bottom, so that's no SAV. But then other locations have SAV, and we record that as present. We also record the depth and the lat and long. We now have a system that has a built-in GPS. So we get a map of true positives, true negatives, false positives, and false negatives. I'd like to thank East Carolina University, the Albemarle Pamlico National Estuarine Partnership, North Carolina Division of Marine Fisheries, the NOAA, and the National Fish and Wildlife Foundation, and North Carolina Sea Grant. All of those agencies help provide funding and support for this project. Thanks for your time. Bye.